Thank you, Sarkis, for the intro, and the organizers for the invitation. Is always a pleasure to see friends and share my science. So my lab is interested in understanding the mechanisms underlying complex behaviors, and traditionally, when we think about behaviors, as Dan pointed out, behaviors that are modulated by both environmental and genetic factors, invariably we think about the brain. But as Sarkis pointed out, we and others, Sarkis and others, we have found, in our case, as you will see, in a serendipitous way, the microbes in the gut could influence the brain in a very powerful way. And I believe this has added sort of a more holistic view of how behaviors can be controlled. So in my lab, we have two complementary lines of research. The first one, more traditional, which aims to understand the mechanisms underlying memory formation. And if some of you are interested, please shoot me an email, and I'm happy to discuss our more recent results. But the second one is a very new one, extremely exciting, perhaps alternative way by which we can modulate brain function. In my lab, we are interested in the mechanisms by which some of those microbes could modulate the brain. So all this started in 2014-15. A grad student in my lab, Shelley Buffington, decided to work in autism. We were interested in autism because there are specific genetic mutations that have been associated with autism, that regulate pathways. They're important, that they regulate pathways that we are very interested. One of them is translational control by the P10 pathway. I invite some of you to read our more recent reports, in which we have identified perhaps different ways that you can modulate the brain and some of those symptoms. In this particular case, developing a very interesting antisense oligo. In this case, in the case of UPF2, in which we delete these genes that regulate a process which is called nonsense-mediated decay, triggers a massive inflammation. And now if we were to use molecules that decrease or dampen inflammation, we can reverse some of the symptoms, including language. But as Dan pointed out, also what we know is about those genetic components, but there are also environmental components, as who mentioned on Sarkis. One of them is the maternal immunity model. But Shelley was thinking about being a mom, and she focused on diet. And the reason why she focused on diet, on maternal diet, is because maternal obesity has been shown to increase the risk of ALD. And those are epidemiological studies that happen in humans. So in order to study this, we use a model. This is the best that we can come out with as a field, a model in which moms were given a high-fat diet. And we put a male, and then we measure the behavior of the offsprings. And we use, as Leticia and others have described before, this three-chamber task. In this particular case, this is the only social task that I will show you. We have three different tasks in which the animal have to decide to associate either with an object or an empty cup or a mouse. And if you are a control mouse, you prefer to interact with the mouse. This is the interaction time. But if you come from moms which are obese, now you can see that the animals are unable to differentiate between the object or the empty cup and a mouse, as a clear indication that they have a deficiency in this task, which is called sociability. Now, and this is what serendipity stroke, when animals were weaned. One of the maternal half-fat diet animals were put with three of the control animals. And when we measured the behavior of those animals, we found that those animals, their behavior was completely reversed. And we could change the ratio. We can do a 1-to-3 ratio, a 2-to-2 ratio. Invariably, every single time that you have a control mouse, which is co-housed with the maternal half-fat diet animal, now the behavior is reversed. So we thought about it, for months, trying to figure out why that was the case. And of course, we know that diet is the more powerful way to affect the microbiome. And we postulated that maternal diet will change the microbial ecology on the offspring, something that happens in humans as well. And this will lead to a decrease or elimination of a subset of bacteria that are required for those social deficits. And because the animals, they are coprophagic and they eat each other poop, the bacteria that is missing in the maternal half-diet animal now will be provided in this passive way when the animals are cohabitated with the control animals. We did 16S ribosomal sequencing, and we could hear each of the dots represent feces for a specific, for an individual mouse. And you can see that you have a clear distinction in the phylogenetic profile of the control and the maternal half-diet animals. And now the animals that they were cohabitated or co-housed with the control animals, instead of being in this cluster, they completely shift to the control group. And that gave us hope that perhaps our hypothesis was correct. But this is just a correlation. When we look at causal evidence, my colleague Sean Cryon at Cork have shown before that animals that they have no germs, they are socially deficient. When we transplant the poop from the control animals, from the maternal regular diet animals, we are able to reverse the social deficits. But when we transplant the poop from the animals that they lack something or they lack a subset of bacteria, because they lack a bacteria which is required for the social behavior, we are unable to reverse the social deficits in those animals. Again, demonstrating that the microbiome was at play. So we sequenced deep and we identified different species whose abundance are altered or reduced in the maternal half-diet animals. The one that exhibits the major change is that bacteria strain species, which is called Lactobacillus reuteri, which was reduced by more than 9-fold in the maternal half-diet animals. If we go and we add these bacteria, this is the maternal half-diet offsprings, again, their behavior is impaired, they are socially deficient. But if we go and we add these bacteria, one bacteria, we're able to reverse the social deficits in those animals. But there's no any bacteria, because if we take a cousin of L.reuteri, L. johnsoni, we find that essentially johnsoni fails to reverse the social deficits, demonstrating that there is some specificity or something that confers this bacteria the ability to do what it does. And I will get that at the end of my talk. So how does reuteri in the gut is able to promote social deficits in the maternal half-diet offspring? So this is, I have no time to go through all data, but let me tell you what we discovered. So inspired by a paper from Susan Ehrman, who has found that reuteri was able to promote oxytocin, we discovered that in the reuteri, what it does is promote oxytocin in the paraventricular nuclei of the hypothalamus. And we discovered that there was a connection between the paraventricular nuclei and the ventral tegmental area. And when we put animals to socialize and we put electrodes in the ventral tegmental area, this is an area that sends reward when you take a drug of abuse or when you have a raise in salary, reward when your paper gets accepted, when you have sex, those neurons will fire. And what we discover is when we put animals that they have never seen each other before to interact. When we measure 24 hours later, we could see essentially a change in the strength or connections between those neurons. And what to me represented like now social interaction became a rewarding experience for those animals. Diet, what it does, it acts as a negative selective pressure and eliminates reuteri from the population. Decrease it 9-fold from the population. And as a consequence, those neurons in the paraventricular nuclei, they are not able to produce oxytocin. And now when animals are put to socialize, the circuitry that sends reward is not activated. And I believe that this might be one of the reasons why those animals don't interact anymore. Why the heck interact if interaction doesn't give you pleasure? But now if you were to put reuteri back, obviously you can turn the circuit on, and now you not only you can turn on the circuit, but now the behavior is elicited. after our paper was published in 2016 in an extremely elegant work, the lab from Bob Malenka at Stanford, have specifically activated and inhibited that circuitry, and they have discovered that they can enhance or impair social behavior, indicating again that this is one of the major circuitry which are driving social reward, and they are making, in our point, they are making the social experience meaningful. And as you know, patients that they have mutations in oxytocin receptors, they have been associated with autism. In animals, in moms, in which you remove the oxytocin, they are bad moms and they don't take care of their offsprings and they are socially impaired. So we believe that the bacteria is driving this very conserved pathway that is crucial for social behaviors. Okay, so now this model is much less popular than the MIA model. I have to say the MIA model was not always popular, but has been characterized by Paul Patterson, Sarkis, Jun, and many others now. So this is, we started this maternal half-diet model to look at neurodevelopmental dysfunction. And one of the questions that I have as a PI was, could this just be in a specific effect in this model, or how generalizable are our findings to different models of autism? The second question that I have is whether the effect is direct or not. I will get into this. And the third one is, what is the mode of communication now? How now essentially those changes in the gut could promote oxytocin in the brain? What is the way that the gut is communicating to the brain? So as Dan pointed out, mice are not humans. And one of the critiques that some of those models have had in the field is that they are not tremendously reproducible, not all of them. So at that point, what I did is I called Shaker and Crowley. Shaki is one of the experts in social behavior. She has characterized dozens of models of autism. And I asked Shaki, what is the more reliable genetic model of autism in which we could see social deficiency? And Shaki's answer was the SHANK3B model. So SHANK3B is a postsynaptic protein, and SHANK3 has been associated with autism. And models have been generated in patients. You have mutation only in one allele. In our hands, the heterozygous animals, if we were to do 3 cohorts, only one of the cohorts show behavior. But if you were to delete or remove both alleles, you have a knockout instead of a het. You have a very reproducible and reliable impairment in social behaviors. So Martina in my lab went and studied this model. And very surprisingly, she discovered that there was a change in the microbial ecology. I don't know, I think Sarkis can talk to this. The microbiologists say that we cannot call it dysbiosis anymore. We need to call it changes in microbial ecology or microbial composition. Still not sure why that is the case. But this has been characterized by a decrease in root levels. When we measure root levels by QRT-PCR, we find that root levels are reduced in this model, indicating that somehow this is a sensitive bacteria, can be affected by an environmental insult, but also can be affected by a genetic insult. Just keep in mind that when you have a somatic mutation, the somatic mutation not only happens in the brain, but happens in every single cell of your body, including cells in the gut or cells, immune cells, which a bunch of them resides in the gut as well. So, but the more interesting aspect is when Martina went and she gave Reuteri to those animals, she found that now their social impairment is actually reversed, either in this sociability task that I described before, or in this so-called reciprocal social interaction that I think has been described before. It was extremely rewarding to see that another group in Israel were interested sort of in the same questions that we were, namely, they took the Shang-3B model, and they also found that reuteri levels were decreasing in the Shang-3B model. And now when they give reuteri to this model, they also are able to reverse the social deficits, indicating that the findings that we have might be, reproducible across different laboratories. And I have to say that apply to at least 4 preclinical mouse models of autism. So happens across different models and also across different laboratories. Something that I think is essential for the development of this sort of nascent field in which data are reproducible across labs. This is an example of how the experiment is done. When we look at the social interaction and we put an electron, we measure the insertion of those receptors, of AMPA receptors in the postsynaptic cell. Those are the receptors who will promote essentially the ability of the neurons to strengthen their connections. When you have a social interaction in the control animal, you essentially have this increase in synaptic activity, transmission, or potentiation. But now, if you, in the Shang-3B model, you prevent this doesn't take place, or actually it does, but the only way that it can take place if you were to add reuteri, or if you were to treat those animals with reuteri. In order to provide causal evidence that those oxytocin dopaminergic neuron was at play, what we did is we remove oxytocin receptor in dopaminergic neurons. Not surprisingly, what we discovered is that those animals, I'm sorry, this is my dog. So not surprisingly, we discovered that those animals are socially deficient. And now when we take reuteri, when we take reuteri, we find that reuteri fails to reverse the social deficit and also fails to rescue those changes in synaptic potentiation. This is a question that I asked to Leticia, and the reason what I asked Leticia is because if we look at the social induced changes in synaptic potentiations are impaired in those animals, but if we were to give cocaine to those animals, now essentially that circuitry is not affected, and we see the potentiation that one would expect when you give a reward, indicating that there is specific deficiency in the social circuitry. As Dan pointed out, perhaps the specific circuits are crucial in this pathology. Now, is the effect of reuteri direct or not? This is crucial because, perhaps reuteri is producing a metabolite which is required for social behavior. So that metabolite, as in the case of last talk from Sarkis, this metabolite can get into the blood and then directly affects the brain, perhaps. But the other possibility is that metabolite is required, say, by this guy here in blue, and in the absence of reuteri, the metabolite is not here, so blue is not here. And this is the one which is the major driver. Obviously, from the therapeutic standpoint, you want to know whether who is the chief in town, is this guy or the guy in blue. So in order to do this experiment, we took germ-free mice, and when we give germ-free, essentially reuteri, in the germ-free mice, obviously there is no blue, there is no green, there's absolutely nothing, there's nothing that we can detect. And now when we give reuteri, we are able to reverse the social deficits, and I know that Sarkis has had a similar success with similar experiments as well. So what is the mode of communication by which L.reuteri restore those changes in the brain? So one possibility, as Sarkis and Leticia pointed out, is the so-called leaky gut hypothesis. The second possibility that we measure is whether the immune system is actually, which could sense changes in the microbial composition or ecology, and as a consequence could produce cytokines that will get into the brain. And obviously the third one is this vagus nerve that in the last, I'd say, 3-4 years has get a lot of momentum, and I think it's an extremely interesting way of communicating not just the gut, but also other organs with the brain. So we couldn't see changes in molecules that would potentially affect permeability in this assay, as Sarkis pointed out. In our case, we don't see changes in the Shankly-B animal. We decided to do a very painful experiment. We thought that perhaps B and T cells were involved in this process. So we crossed our animals with the RAC2 knockout animals. The RAC2 knockout animals, they are socially normal. But when you cross it with the Shank3B, which are socially impaired, this is what you get. And now when you take Reuteri, Reuteri is able, despite the fact that you don't have mature B and T cells, Reuteri is able to reverse the social deficits, and is also able to promote those changes in oxytocin and plasticity, indicating that at least mature B and T cells don't seem to be a primary driver or mechanism by which Reuteri drives this. Whether the innate immune responses at play is something that we don't know, but we are currently investigating. The third possibility is the vagus nerve, and we have mastered this so-called technique called vagotomy, where you just interrupt the connection between the gut and the brain, and you find that this is the sham animals. When you cut the vagus nerve, Reuteri fails to reverse the social deficit. Some of you may say, "Oh my God, this is an extremely aversive task. This is such an aversive task that the animals cannot perform the behavioral task." The answer is no, because if we take the same animals which are vagotomized and now we puff oxytocin intranasally, now we are able to essentially promote social behavior, demonstrating that when we interrupt the so-called via of communication between the gut and the brain that Reuteri use, namely the vagus nerve, that's why the bacteria has no effect. Okay. As we speak, we are actually doing recordings where we record the vagus and we are using different bacterial composition. And I'm happy to say that we are able to separate different signals that come from the vagus. So the vagus sends a bunch of other stuff. And now the goal is try to deconvolute a chemical structure into a synaptic readout that perhaps could help us or could educate us to see how essentially different chemicals, in this particular case, microbial metabolites and different bacteria species can activate the vagus and talk to the brain. So my lab is very much mechanistic. I think this is where the field should go. We have heard in the past as different bacteria species that could be associated or could modulate a specific behavior. Most of the focus of the neuroscience field, people working in neuroscience, has been in trying to identify cell types and circuits and receptors in the brain. We believe that we have identified a circuit and receptors which are important. We don't know in terms of cell types. We believe that the dopaminergic neurons in the VTAs are key, but we don't know whether this is the only structure or the only cell type and structure that is driving this. So there's much to be known. The vagus nerve seems to be the major driver by which this particular bacteria species. We have other bugs that do some other stuff that the vagus has nothing to do, but in this case, the vagus seems to be involved. Whether there is a role for the immune system still remains to be seen. The major question for us is molecules which are either produced by the bacteria or they are induced as a result of the bacteria being in the gut. And I have no time to share that with you, but we have discovered that the bacteria is able to induce a particular metabolite, and by just providing that metabolite to the animals, we are also able to reverse the social deficit. So obviously, there's a lot of hope on what we do in animal models. In reality, I have no idea if anything that we are doing here may have any potential for humans. So if you have a mouse and you have a mouse which has autism-like symptoms, we may have something for them. But as I said, we don't know whether this would be translatable into humans. That said, I think it's extremely important to consider to start to determine systematically objectives and criteria when we get into this new era of perhaps microbial-based therapies. And when I say bacteria strains, of course, this is applied to also bacteria metabolites, right? So I think it's extremely important that this doesn't happen once and doesn't happen in one model. I think the robustness of the technology that one is able to generate has to be applied to some models, perhaps not all models, but perhaps two models, another two, it doesn't work. So we can, perhaps this is going to be a way to personalize medicine, right? Specific population or stratified patients that could be more susceptible. But I think it's important that happens in multiple models. It is key to determine whether this microbe is the one which is driving this or metabolite, and if it's an indirect, you should maybe go for the indirect microbe. I think it's extremely key to think about the mechanism by which the microbe could do this. In this particular case, clearly our microbial treatment or microbial intervention, it is clearly affecting oxytocin. So if I were to think about an effective clinical trial, I will go with patients that have been responsive when oxytocin was given to them. Oxytocin, just keep in mind, oxytocin has been given intranasally. In some cases have been shown success in terms of social behavior, but the dose that one gives is super high, as Dan has shown in animals or other people. The effect in animals lasts, say, half an hour. By 24 hours, the effect is completely gone. So we have an interest to have something that will promote the endogenous amount of oxytocin. Reproducibility across laboratories, I think, is central, and that has been shown for this case. And of course, obviously, the microbial treatment that must be used, or the microbial or metabolite intervention that must be used, has to be safe. And those are some of the criteria that we discuss in our last neuron paper, as a way to just to open, not like rules that one has to take, but just to open the discussion with people like Sarkis and many others which are leading this, on to design ways how to go about. I think it is a lot of hope, but I think explicit criteria should be defined. So with that, I would like to thank everyone who is in blue here. I have an amazing group of people. Half of my lab is working now on so-called microbiome gut-brain axis. My collaborators on that respect, Rob Britton and Shaw Petrosino, and I will be happy to, my funding agencies, you guys for the invitation. I'm happy to answer any questions that you guys might have. Thank you very much.
Thank you, Mauro, for very elegant and very mechanistic work, as you mentioned, which I do agree the field needs more of. Maybe that's true for all fields. There's one question that came in that I think you can answer fairly quickly. The question is, did L.reuteri affect the composition of the microbiome? I have read that L.reuteri also acts as an antifungal. Now, you showed us evidence that the organism works in germ-free mice, but is there even a potential that it may be working through remodeling the microbiome?
Well, so when we did, this must not have been the best approach. If you were to do a 16S ribosomal sequencing, in both, in the shunt-free B model and the maternal hepatitis model, adding of reuteri does not significantly convert the mutant microbiome into the wild-type microbiome. So we don't see major changes. Whether it's going to be specific changes, we didn't have the resolution to look into this. And based on the experiments that you pointed out, we find very unlikely that this is the way that the reuteri is working by modulating the microbiome. In this work, which is now under revision, I mentioned that there is a particular metabolite that is induced as a consequence of the presence of reuteri. And if we were to give that metabolite alone, even in germ-free mice, we are able to reverse the social deficit. Something extremely important to point out here is that unlike some of the experiments that others have shown, this is specific. The only thing that we can reverse is the social deficit. If we look at hyperactivity and so on, we are unable to reverse those changes. So perhaps Dan was talking about following now the cancer field. And the cancer field, something which has been very successful, is a combination of therapies. The way that I see this is that perhaps these microbial treatments could be combined with more canonical, treatments that affect directly the brain to affect different symptoms. So no, I don't think that all symptoms are necessarily microbial driven.
And I think we must move on, but I think having a very robust effect on social behavior, not other behaviors, may actually be a good opportunity for you to dissect the circuits, right? Because it may be more straightforward to identify which circuits are meeting one behavior as opposed to multiple behaviors.